Good morning, everybody. I understand I'm between you guys and coffee break, so I'll try and keep this brief. I only have one slide today. I'm just going to give you an overview of the company and the history and what we're about at Tmunity. Thanks to the organisers at Azam for allowing us to present today, our first investor relations presentation actually for Tmunity. So Tmunity is an end-to-end stage T-cell development company that has a vision to help transform the practice of medicine by bringing forward the next generation of T-cell therapies, be those in the form of T-cell receptor technologies or CAR-T technologies. We're a company that was founded out of Penn back in 2015. We have 6 scientific founders: Carl June, Bruce Levine, Yangbing Zhao, Jim Riley, and Anshu from Penn. And we also have a non-Penn founder, Bruce Blazar from the University of Minnesota. And between these individuals, I think we have probably the best expertise in the T-cell spectrum in terms of science translation and the ability to actually manufacture T-cell related products, and obviously a track record of having helped bring the first CAR-T therapy over the finish line in last year in August when Kymriah was approved. That obviously was a technology that Novartis developed in collaboration with Penn. Penn have seeded many companies over the years, but it's the first time they actually invested in a company as well, and they placed a $5 million seed into the company. This was matched by Lilly Asia Ventures, who also contributed $5 million, and that actually set the company up well in 2016 to start to really invest in novel and accelerated science in the cell and gene space. So probably one of the first landmark events that happened that I don't think a lot of people were aware of in terms of the relationship with Tmunity is that back in July 2016, June-July, Carl June and Yangbing Zhao presented at the RAC committee where they demonstrated the ability to actually gene edit a T-cell, remove the endogenous alpha and beta chains, and also knock out PD-1, in effect, for the first time demonstrating that you can actually apply CRISPR to a direct therapeutic setting and actually create a manufactured product. And that was obviously something that a lot of people took note of. What folks didn't realize at the time is that Tmunity has access to this product as part of its portfolio in a collaboration with the Parker Institute. In October of the same year, we leased out our manufacturing facility. I think as investors and analysts in the field, you're probably getting a theme that in this space, unless you have access to and an ability to embed your own manufacturing processes in a GMP setting, you're left at a competitive disadvantage stage later downstream. So we made a bold decision to lease out our own manufacturing facility, and we were extremely fortunate that there was a facility in northern Philadelphia that formerly had been created by a great company in its time, Tengion. It was one of the first sort of vanguard companies in the regen med space that unfortunately did not achieve success. But I give those guys a lot of credit because they built a beautiful GMP scale facility that Tmunity is now privileged to have in its possession. It's a 46,000 square foot facility, which we can triple in size if required as well. And we're actually in the process now of reactivating that site, and I can talk a little bit about what we're doing there. Towards the end of the year, in 2016, we submitted 2 INDs to the FDA. One was the triple knockout T-cell engineered CRISPR-edited product that I spoke about, and we also submitted our other IND in relation to our first CAR-T program in prostate cancer, the so-called PSMA target, prostate-specific membrane antigen target, and those INDs went in. I had the great pleasure to be appointed in December of that year. In early 2017, my chief business officer, Michael Cristiano, joined. And recently we just announced Jason Krentz as our head of manufacturing and chief technology officer also joining. We received additional funding of $10 million by Lilly halfway through the year. We progressed our clinical programs, dosing our first patients in the PSMA CAR. We're now actually in the clinic with our CRISPR-edited program, actively screening patients and hopefully treating them. We then received additional funding from the Parker Institute as part of a transaction at a seed stage. And very importantly, we completed a transaction with the University of Pennsylvania in an exclusive license agreement, which was building on the original agreement that was established when the company was conceived in 2015. And you probably all saw that we were successful in securing our Series A of $100 million in January, third week. I'm pleased to announce that this will be extended to another $35 million, and we'll be announcing a US investor later this week, and we will consider this Series A closed and capped at $135 million. And we are fast moving forward in the clinic now with the current programs, and we plan to have additional INDs going into clinic later in the year. So thanks for the interest in Tmunity, and happy to take any questions you have today.
Yeah, great. So, if we think of first generation CAR T, primarily focused on CD19, where can you take your programs?
So the first generation of products, this was it's a transformative moment for the space, and I think those of you who listened to Kathy High this morning can testify you're building off the shoulders of giants, right? This is something that took 30 years in the making. With folks like Carl June and others working in this field, initially in the HIV space, and then having the vision to do something in B-cell malignancies. And success breeds success, right? So people think it was an easy journey. I can tell you, having been involved in a small part in that journey, it was not easy. I think now the vision is that, that was a jump to the moon. And I think companies like Tmunity, we want to go all the way to Jupiter and Neptune, Pluto. And it's not going to be easy. But, the vision now is for us, we are tackling solid tumors. We're tackling more aggressive liquid tumors as well. And it's not going to be as simple as a CART construct attacking a specific antigen and fingers crossed and hoping for the best. Even though that is a huge monumental achievement, that's going to work well in B-cell malignancies, but we also understand that the hurdles for solid tumors is going to be very different. So how do you actually understand not just engineering and making those T cells more powerful, but how do you manipulate the environment around a tumor? And I think that's what companies like Tmunity and every other company that's in this space is trying to master, is that as well as killing the cancer cell itself, how do you then create an ecosystem around that cell that either inhibits a growth factor? For example, if you look at our PSMA CAR, it's not just a CAR attacking a antigen in the form of PSMA. It actually has a switch developed into it that tackles TGF-beta. So transformational growth factor beta is highly overexpressed in certain forms of prostate cancer, PSMA being one of them. We've developed a technology that allows you to shut down TGF-beta locally. So as an example, right, that's the kind of approaches that are going to happen. It's going to be combinatorial therapies, not just, adding a PD-1 in with a CAR T. It's going to be about understanding how do you engineer those cells, now that we have the tools and technologies to do them, but also address environmental factors. And I think that's going to apply to hematological malignancies and also clearly to solid tumors.
So in terms of your initial selected targets, what about those prostate, melanoma, what about those and their specific microenvironment make them good initial targets to have proof of concept of the technology that you're bringing forward?
It's a great question. So let's take NY-ESO-1. So NY-ESO-1 is a very old, tried and tested target. Cancer testis gene, everybody has looked at that target. So the reason for picking that target in particular was actually driven by safety and something that is very well characterized. So this is hopefully going to be the first evidence in a human setting in the US that CRISPR technology can work. In order to do that, you can imagine regulators set a high bar from a safety perspective. So the real reason for going with a target like NY-ESO-1 is that it's prevalent in myeloma, melanoma, but more importantly, it's a safe and validated target. And if you attack that target, you've probably got a good chance from a safety window perspective that you're going to succeed and generate data. And we hope we see some, obviously, clinical benefits as well. So there was a scientific and I would say regulatory science rationale to a target selection in the case of our first TCR program. On the PSMA front, this was a hugely exciting target. It's a huge unmet need area. There have been a number of trials conducted in the space but have not been successful, and partly because of this inability to really tackle beyond the cell itself, as I mentioned before, the tumor microenvironment. And we felt that this was a patient population we could get to quickly, and as importantly, the science was established enough that we could take a chance on tinkering with that system quite relatively easily. And as testified, with the IND that was submitted, we were able to get into the clinic in relative short order.
Beyond the CRISPR editing that you're utilizing, is there other advances to your therapeutics that you're also implementing? And then in doing that, what's the risk that as you move into the clinic, being able to ascertain if one or another is causing benefit or perhaps a negative effect?
Yeah, so I think it's a great question. So for us, it's not just about the targets. It's about creating a stable manufactured product at the same time. So within our model, we have over half a dozen licensed programs now from Penn. We have multiple shots on goals that we've initiated in terms of getting the best in vivo, in vitro data developed as quickly as possible to seeing which are the strongest horses that emerge and get into clinic. So that's one key component. But I think really to underscore your question, it's really about that ability to tie in a target characterization very early on and translating that to manufacturing findings. So I personally believe this space is going to be won or lost, not just purely on the scientific story around a tumor type being attacked and overcome, which is clearly very important, but it's about the ability to create a meaningful manufactured product. So we are spending a lot of time and a lot of resources and capital on really addressing that translational need. So let's face it, the first generation of products that are coming through now, be there with the first generation CAR T therapies and that are being planned, require a lot of work, right, in terms of optimization of process. So for us, it's very, very key that very early on, as we identify a target when we're developing that, in terms of the early stages of manufacturing process science characterization, that we develop a good understanding of what that product needs to look like in the marketplace whenever we're successful. And the timelines are pretty accelerated, right? If you look at, the story of products like Yescarta or Kymriah, these went for proof of concept in 2012 to approved products in 2017. Somebody had told me that in 2012, 2013, there'd be an approved, CAR T therapy within 5 years. I wouldn't have believed it, but it happens. So if you take that and look at the responsibilities that you have as a company, right, to not just promote the science, but actually create a real product that can be scaled. A big effort in companies like ours is to make sure that we've doubled down in that space and the science around enablement. So how do you enrich a cell better? How do you stimulate a cell better? How do you reduce the cost of goods radically to make these therapies scalable? Because as we know, in the populations we're looking at, and we have ambitions to go for some pretty major indications and targets in the future, which I'm happy discussing at a future setting, you need to have this sort of ingrained in your infancy as a company.
Yeah, absolutely. In terms of the therapeutics, do they offer or facilitate any advantage in terms of the preconditioning regimen?
So we are looking at both conditioned and non-conditioned regimens. For example, if you look at the protocols that we have right now for PSMA, our initial safety cohort is in a non-conditioned setting. First 3 patients have cleared the safety hurdle well, so we're now increasing the dose. And then the third cohort will be looking at the pros and cons of conditioning, because obviously we know that right now that is the standard of care. You need to create some compartment for these cells to proliferate that do proliferate very, very quickly. But obviously we're all looking for the right balance of can you get these T-cell products, be there a CAR or TCR, really advancing and proliferating in the absence of conditioning. So it's a bit of a stretch right now, but we are looking at ways through our clinical programs to see the pros and cons of both pathways.
And then just back to manufacturing, in terms of your design rationale, are you considering what today can be done on a fully automated closed system basis? Or are you anticipating some advancements there that you'll be able to utilize, as you get to commercial stage?
So we're looking at both, frankly. So right now, our main focus of the products that we have are autologous products, but we've also got research programs looking at allogeneic platforms. So we have to think fast forward as a company in 5 years' time, what's the landscape really going to look like? Now, there's going to be a backbone of manufacturing capabilities and scientific translational capabilities that will apply equally to whether your product is autologous or, an allogeneic product. So having an understanding of that up front and embedding that in your plans is going to be very key for companies like Tmunity. So the question about closed systems or open systems, I think our focus, first of all, is to take the products that we have and really tweak those to optimize and look at aspects of that can really be part of a contained closed system. And then obviously there's many companies out there, Miltenyi and others, that have been looking at this for a long time in terms of what's this model going to evolve to. So are we looking at an autologous manufacturing centralized setup, or are we looking at point of service care models that could evolve? And you have to be able to play in all spaces, right, as a young company. But for us, the key is to really translate the findings into real manufacturing science capability, that initially we pivot to multi-center studies as quickly as possible, with a view, obviously, to getting this out to patients as successfully as we can commercially in a few years' time.
All right, great stuff.
Thanks for your time.
We'll be looking to see what you put out there as achievements.